Hi, my name is Ilya Nemenman. I'm at Emory University in the departments of physics and biology. I'm a theoretical biophysicist, and I was asked today to give a talk about our recent results on theoretical modeling of neural dynamics. And first of all, I want to thank the organizers of the meeting for inviting me, and thanks to all of the supporting personnel for actually making this happen. This is supposed to be about a 15-minute talk, and we'll see how it actually shapes up in real life. Okay, so with that, what I'm going to talk about is the null models of large neural networks. This is a work which is primarily based on two papers, so the talk will be structured in the following way. First, I will talk a bit about what I think are some of the challenges about building a theory in neuroscience, theory of a brain, and then I will go very quickly through two case studies, two examples where we have been able to develop what I think are better null models than most other groups are using. This work has been done in collaboration with two other researchers. Audrey Sederberg was a postdoc at Emory and now is a faculty at the University of Minnesota. And Mia was an undergraduate student here at Emory and now a graduate student at NYU. So let's start with the following thing. Now about 7 years ago, here at Emory, we organized a computational neuroscience meeting. And in some of the sessions at that meeting, there was sort of expressed a common opinion by more than one researcher, more than one scientist, that the final theory of biological systems will be large multi-scale computational models, and we need more and more experimental data to specify details of such models. And so the final theory of the brain is going to be basically a large electronic brain with the one-to-one accurately simulated biophysical processes. And there's a lot of problems that I see with this statement. So the first one is that what is really the final theory of the brain, right? Do we even need a theory of everything, all of the processes, all of the things happening at the same time in any biological system such as brain, or in fact in any physical system, right? So if I were to ask, what is a final theory of this coffee cup, right, of this coffee mug, you probably would not be able to answer this question. It would depend on which kind of questions you actually are asking the theory to answer. Do we want to understand how quickly this cup is going to drop if I let it go? Do we understand if it's going to crack? Do we understand how the color of change as if I put it on the window sill, and so on and so forth? There is not a single theory that explains everything there is to explain about this cup. We ask specific questions and we build theories for specific questions, right? And so in my opinion, the sort of the paradigm that people have developed in physical sciences, that we do not model bulldozers with quarks, suggests to us that the theories, be that of inanimate systems or of this very complex biological systems, the brain that we're interested in, theories must be based on small sets of degrees of freedom, and these degrees of freedom must be relevant for a limited set of the phenomena that we want to explain, right? We need to know the mass of the object in order to figure out how it responds to a force attached to it, but we don't really need to know all of the details of where exactly this mass is organized, right? And in a similar sense, in the brain, we'll probably need to know some structures of the brain, some dynamics of the brain to give answers to some specific questions, but we will never need to know everything. And so the goal of theory, in some sense, is to find what are these relevant degrees of freedom that we need to identify, to experimentally measure, from which we then can build answers to specific sets of very well-focused questions that are being asked. So there are, in general, many different ways how theory has been used going to leave aside all of the old work from Hodgkin and Huxley and so on and so forth and just focus maybe on the last 5 to 10 years. So what we have understood is that the neural circuits can actually be modeled mathematically in all of the details. There's probably no paradigm change at this level of small circuits. With enough of experiments measuring all the details of this involved cells and their couplings and so on and so forth, and with enough modeling, which could be dynamical system style modeling or stochastic processes style modeling or yet many other different approaches, we can understand this circuits, right? And it could really be that there is no theory in neuroscience, right? We are always forced, or will forever be forced, to build this detailed model, sort of like people were saying in 2014 at the CNS meeting, ever larger, ever more detailed models and populate them with data. And that's possible. Personally, I'm not that happy about that, right? Because I'm not sure what I'm doing in science. And secondly, I also believe it's probably not quite right, because for me to be able to work with my brain, right, for me to be able to understand what the world is around using my brain, there has to be an ability to summarize the details in, summarize the activity in some number of interesting relative degrees of freedoms. I don't have the entire age of the universe personally to learn how my brain works, right? I don't have the age of the universe to learn how to use my brain to control my muscles. If I'm able to operate in the world, it's only because there are these underlying low-dimensional degrees of freedom that are learnable, and that's what we're going to focus on. And so in this regard, the one role of theory that people have focused on is based on various principles, optimization principles, that we need to control our muscles quickly. We need to not use a lot of energy for that. We need to be able to send information efficiently, and so on and so forth. And from that, we can predict the circuit structure. We can understand why the brain is structured the way it is structured. Right? And a lot of other people, I'm sure, will talk about that. What I'm going to focus on is a slightly different question that theory, I think, can answer in neuroscience. So what has become clear in the last decade or so is that measuring thousands of variables about the activity or the structure of the brain is relatively easier than knowing how to interpret these measurements. We can record population activity from nowadays 10,000 neurons, so we can record connections between thousands of neurons. And what do we do with that? What does it mean? When should we be surprised? We sort of understand that more is different, that interesting emergent features are going to show up as you put thousands of neurons together, but our intuition fails when we record from these large populations of neurons, or we measure structures of activity patterns of thousands of neurons, right? We do not know when we need to be surprised. If we get a certain correlation, if we see a certain feature in the data, it may be surprising, or it may be something that is something that one should expect, right? And we don't know when what the boundary between the two. And so what's going to happen in this talk is, even though brains certainly are not random networks, what I will show here is that many seemingly surprising results that people have thought are surprising in the context of traditional detailed specific neural network models, these results are modeled exceedingly well by random networks model of the brain. And so maybe our assessment of what is surprising and what is not should be changed. Maybe our idea of what is a null model of the brain should be changed. Maybe the null model should be just a random network, and we should look for deviations from that. And maybe we should develop experiments that are designed to detect this newly surprising things that we actually know now are surprising. And so if you don't have any more time, this is basically the main message of my talk today. And from this point on, what I will do is to lead you through two specific examples where indeed it's pretty clear that we need to reassess what is surprising, what should be the null model that we're going to be using. And this will all fit into this slide that you are seeing right now. Okay, So the first experiment that the first model that we are going to discuss, this is a beautiful paper by N. Churchland's lab, Najafi et al. In 2020 in Neuron. And so what they did there is they recorded from the mouse brain as the animal was performing an auditory choice task. Right? There are some clicks that are happening in the background, and the animal needs to make a choice as this clicks happening with one frequency or with another frequency, and depending on the number of these clicks, the standard psychometric function, like here on this plot B, which is taken from the Najafi et al. Paper. And then there's a beautiful experimental setup where multiple different colors are used to record different properties, different molecules from the brain as the mouse is doing this recording. And as a result, after a lot of quite ingenious manipulation of the data, it's possible to identify which neurons are excitatory, which neurons are inhibitory, and it's possible to understand what is the activity of individual excitatory neurons and what's the activity of individual inhibitory neurons in this system. And so that's what's shown in figure C here. And so types of question that one can ask, what was the original goal of this paper, was how well can one predict behavior of the animal? Is the animal going to make a left choice or a right choice, right? How well is it possible to even understand, forget about the behavior, is it possible from the activity of the neurons to understand what the animal is hearing? Is it hearing the high frequency or the low frequency signal? And then the general ideas are that excitatory neurons are the ones that code for information, and inhibitory neurons sort of keep the activity to manageable levels, and so maybe one should be able to make these predictions from excitatory neurons, but not from inhibitory ones. And so the question is it really true? Can one make classifications of the stimuli heard by the animal from excitatory neurons or from inhibitory or from both? The main result of the paper was that the inhibitory cells were almost as good as the excitatory cells at making predictions of whether it was a low frequency versus a high frequency signal that the animal has heard. And the paper has developed an intricate neural network model that allowed the selectivity for the high or low frequency signals, both from the excitatory neurons in the network and from the inhibitory neurons in the network, and from both. And so when we thought about this, we said, can we try a different null model, right? So what I mean by this is that instead of looking at very structured models where specific excitatory cells talk to specific inhibitory cells, and there are pools of those and pools of this, and they're cross-interacting, etc., and you need to add more and more details to this specific couplings in order to actually explain what you see in experiment. Maybe we should start with a different site, not with a structured network being the starting point, but with the most unstructured network being the starting point. And so what we did is we developed a random recurrent neural network model where you have the pool of blue excitatory neurons which talk to each other, different sizes indicate their activity. There is a pool of red inhibitory neurons which talk to each other and talk to the excitatory neurons. There are input signals which project to both in excitatory cells and inhibitory cells, and we of course tried input signals projecting only to excitatory and only inhibitory and all of modifications you can think of. And then this network sort of does some kind of dynamics, and then after that one tries to identify whether the inputs were high or low frequency of clicks based on some kind of linear nonlinear classifier readout from the excitatory neurons or inhibitory neurons or all neurons. And in this subsequent slides, blue is always excitatory, red is always inhibitory, and black is always excitatory and inhibitory. This is similar to liquid state machines architecture, except that we're not fine-tuning this architecture in any way, which is what's required for liquid state machines. This is just plain vanilla neural network in the EI balanced state. Okay? And so once we did that, then we could do all the same recordings from these networks as was done from the mouse, right? We could try to understand if excitatory neurons and inhibitory neurons are selective for high or low frequency stimuli, to which extent are they selective, at which point in the dynamics of the network they become selective, how good the selectivity is from either the excitatory neurons or inhibitory or from both, and so on and so forth. And I'm just going to show you a small subset of results that we found. So, for example, one could ask in many different instances of this network realizations. We're having here 14 different realizations of such networks. What was the selectivity of individual excitatory cells or inhibitory cells for either the high or the low frequency of clicks, right? If I look at an individual cell, excitatory or inhibitory cell, will I be able to say, even with small probability, but not a 50-50 probability, maybe it's 51% versus 49, can I say whether the network was excited by the low or high frequency of clicks? And so what we see here, this is on the figure on the left, again, blues are excitatory, red are inhibitory. There's essentially no difference between the selectivity that emerges in this network from excitatory and inhibitory cells. And the selectivity emerges because, even though it's randomly connected to large networks, there are some fluctuations in the sense that, this particular neuron got connected to that one, but not this one, with such and such strengths, but not such an, another strengths. And overall, there is going to be some small asymmetry. This a particular neuron is going to be a bit more responsive to high frequency stimulus or low frequency stimulus. And then you see that the selectivity is actually not that high, right? It's in the ballpark between 0.5 and about 0.1 accuracy. This is the actual metric here is the area under the curve above 0.5. So these neurons are 0.6 area under the curve selective. And if you look at experiments are all in the range between 0.1 and 0.16 for the neurons that they're recording from, right? So the most selective neurons are exactly in the same range as we are recording in this simulated neurons. And then you put them all together, and even though individual neurons are only weakly selective, but when you put hundreds of them together, you can easily train a classifier that is going to predict whether the animal has was just experiencing a high or low frequency of stimuli. And this is what the plot on the right shows. Again, if we try to train from all of the neurons, excitatory and inhibitory, we do slightly better than if we train from excitatory or inhibitory neurons only, but there is no statistically significant difference between excitatory neurons or inhibitory neurons in representing which stimulus the animal is experiencing. And the numbers, the ability of this network to classify whether it was high frequency of clicks or low frequency of clicks, we're recording numbers anywhere in the ballpark between 0.7 and 0.85, with maybe one exception for network number 6. And then the experiments are also recording the same accuracy, 0.7, 0.85, right? So this random network model does not just qualitatively match what the experiments produce, but it quantitatively matches what the experiments produce to within 1 or 2 significant digits. And so the statement here is that all of the experimental results that were reported, essentially all experimental results that were reported, we were able to reproduce with the random network model that I presented, and they matched the theory and the the experiments matched each other to within 1 and 2 significant digits. And so this emergent selectivity, which is nearly equal for both E and I cells, is expected in a random network. It's not an unexpected result that requires fine-tuning and design of a network in a very particular way. No, you expect it in a random connections. And we can make predictions here. In a random network, changing stimuli will change the classifier support very drastically, but in a precisely designed network, that's not necessarily the case. So the changes to classifiers based on changes in the stimuli might actually explain or might actually say whether we are whether the model of the random network we have developed is actually the right model of the experiment. In order to do that, what one needs to be measuring is stability of classification, multineuronal correlations, not just pairwise correlations that were measured, and so on and so forth. But it looks like this very simple network is at least a good starting point, a good null model for this type of experiments, and we need to build on top of that. Let's switch gears a bit, and we are now going to look at the mouse in a virtual maze experiment. This is a work by David Tang's lab. Louis Michelin is the first author of PRL paper in 2020. So what's happening there is we have a mouse in a virtual maze. It's running through the maze, and activity of neurons in hippocampus is recorded. And the question here is actually a very exploratory question. What are the interesting correlations among these neurons? If I try to build a coarse-grained, large-scale model of this neural activity, are there any interesting correlations that are emerging that are surprising? And so in order to answer this, what the researchers have done is basically they look, as in picture B, they look at every individual neuron, they find neuron pairs that are the most correlated with each other, and they just sum them together, okay? And so this produces sort of a hyperneuron, and this is very similar to coarse graining that people use in statistical physics. The idea is that we are going to combine together neurons that are similar to each other, the most correlated. So we're not throwing away much of information because they are strongly correlated with each other, and we get this a collective neuron, a hyperneuron, and maybe those hyperneurons would be a good enough description of the underlying neural dynamics. And so an interesting result emerges is that if you start looking at various correlations, be they spatial, temporal, and so on, correlations between these coarse-grained merged neurons, then the correlation functions as the measure of the, let's say, temporal distance, or the correlation function distance between the time points in which you are calculating it, start developing beautiful power laws. And we all know that power laws are not very common to get in physical systems, in an inanimate system. So maybe this should be something surprising that needs to be explained. So here is a model that we developed for this. We have a network of neurons. They're not talking to each other at all. They're just on or off neurons. And there is a series of about 5+ latent variables. And these latent variables are randomly connected to the neurons. Latent variables undergo some kind of random walk dynamics, and they drive the activity of the neurons. And we're going to run the same type of the coarse graining merging neurons together as the Michelin et al. Paper has. So this is what we get. If I look, for example, at the variance of activity of these hyperneurons, and I plot it versus the cluster size, sort of the number of neurons put together into one particular cluster after the coarse graining. So if neurons were completely independent from each other, you add them together, we're going to get the law of large numbers, the central limit theorem, and the variance should grow as the number of neurons in the cluster, like number of neurons combined together. And in fact, we don't get that. We get the variance which grows with the power law of 1.367 plus minus 0.009. And in experiment, when they did this type of analysis, they got 1.4 plus minus 0.006. It's a perfect, nearly perfect agreement. Let's keep figure B because I'm running out of time. For example, in figure C, what's happening is that we are looking at this activity of these big clusters, of 100 neurons put together hierarchically, and I can calculate the principal components, get the eigenvalues of activity of these clusters, and plot this the size of the eigenvalues versus their rank versus their number. And if you see, there is a power law there. Experimentally, 0.71 plus minus 0.006 was measured, and we are recording in figure C from this very simple simulations 0.65 plus minus 0.009. Okay, things match to experiment in theory match with each other at 2 significant digits level. Even more interestingly, one can ask, if I look at the activity of these clusters as I get neurons together into each other, the activity should become, it's a sum of many variables. Variables are not quite independent from each other. If they were fully independent, then the activity of individual cluster would just be a Gaussian distributed variable by the central limit theorem. But neurons are not independent. They're not fully dependent on each other, but they're sort of somewhat dependent. And so maybe I will not get a Gaussian distribution, but I will get a distribution that looks nearly Gaussian, right? Or maybe not nearly Gaussian. Maybe it's going to be away from a Gaussian. Let's see what's going to happen. And so we do this in our system. A Gaussian here would be this dotted line that you see in the middle. And what you see is that as we coarse grain the system, we combine neurons in groups of 16, 32, 64, 128, you start seeing that there is some kind of a limit, right? The yellow and the orange lines are essentially on top of each other. So we reached the limit and we didn't reach a Gaussian. So the activity of these neurons is on large scale very non-Gaussian. And while I'm not showing this here, this plot is quantitatively matching exactly what was recorded in the Mishralam et al. Analysis of the experiments from Tang's lab. Not qualitatively, but quantitatively, and I want to stress this. And so, as a result here, essentially all reported experimental results were reproduced by our model within about 2 significant digits within this null model. We have no idea what those latent variables are that are needed to drive the active neurons. And in some sense, the next step of this type of analysis, next step of such theory should be to identify these latent variables, and maybe those latent variables are precisely the relevant degrees of freedom on which future theories of this particular part of the brain need to be built. I don't know that. But what's clear is that we should change what our null model is and what our expectations are about what's surprising and what's not surprising in such experimental recordings. Power law scalings are not surprising at all in model with latent degrees of freedom, and it looks like they match the experiments with very high accuracy. That null model is probably quite reasonable. What it also means is that we need to be designing experiments and analysis not to identify those power laws in the future, but try to identify deviations from those power laws. Right? We need to measure higher order correlations to see whether they are meaningful or not. We need to make measurements with and without perturbations to the brain, because it turns out that responses of different null models to perturbations are actually quite different, and that would allow us to make distinction to understand whether the prediction, the model that we have built here is actually the right model. And so, as a summary from all of these parts of the talk, I want to say again that I don't believe that the brain is random, right? By no means. But random networks are probably a good starting point, a good null model for building theories of the brain. And it's very different from what people normally do in modeling brain activity. And with that, I again need to thank the organizers. I need to thank the people who have contributed to this work, Mia and Audrey, and I'm going to stop here. Thank you.